Hello, welcome to the lecture, and I'm Thomas Ming-Shui Chang. I've been working in artificial cell for more than 60 years. It's a pleasure to briefly summarize this area for you. First, let's ask the question: is it possible to prepare artificial cell consisting of monothin membrane with inside separated from outside, but also outside cannot enter? Small molecule, however, can equilibrate very quickly to be acted on by inside and come out. The first simple one prepared consists of hemoglobin inside artificial cell. It binds oxygen and releases oxygen in a way very similar to red blood cell. Now, after medical school, I went back to work full-time and become more ambitious and asked the question: red blood cell consists of membrane enclosing hemoglobin and lots of enzymes. Is it possible to prepare the same artificial cell except changing the membrane to an artificial membrane? These are the artificial red blood cells that are exactly the same in content as red blood cell, except the membrane is now nylon and cross-linked membrane protein. The hemoglobin and red blood cell enzyme is retained inside, separated from outside, but substrate can go in, acted on, and then come out. These are very thin membranes, so when you break it, it becomes shown here as ultra-thin membrane here. It's very thin and very permeable, so if you put it in hypertonic solution, it shrinks as water comes out, and if you put it back in hypertonic solution, it retains its shape as water re-enters. You can change the membrane, making it with lipid complex polyhemoglobin. You can also put in passive transport mechanism. You can also put in active transport mechanism. So therefore the answer is yes, we can prepare a complete artificial red blood cell. But can we use artificial to innovate, to translate? So let's ask the question: can we vary the content, size, and configuration? The beginning attempt and innovation is done whereby artificial cell of nanothin membrane can be entrapped with cells, hemoglobin, enzyme, magnetic material, adsorbent, and other things. It's separated from outside, molecule can go in, acted upon inside, and then come out. Now, going further now, at present, you can in fact put in all sorts of material, artificial, biological, or active material, as you can see. So there's unlimited content. And you can also vary the configuration from micron dimension under different names. People like to give different names when they extend it for different uses. You can make it macro when you put in cells, and adsorbent. You can make nano dimension. People call it nanoparticle, nanocapsule, liposome, and others. You can make it also in the form of nano biotechnological soluble complex, for example. In my 1972 monograph on artificial cell, I emphasized that artificial cell is just an idea. Much physical configuration are possible. Indeed, in the last 10 years, there has been extensive increase in configuration as extension, improvement of the idea are being made. within a few more years, it's expected that much more configuration would be possible as the area is extended and improved. Thus, indeed, content, cell size, configuration can be varied quite extensively. But the question is, what can you do with all these? This is an example of the possible uses. I'll let you look at it. Microdialysis, drug delivery, blood substitute, biotherapeutic, vaccine, cancer therapy, immunotherapy, enzyme gene therapy, cell therapy, biotechnology, nanotechnology, regenerative medicine, agriculture industry, nanocomputer, nanorobotic, nanosensors.